reason why indirect immunofluorescence is better is because the second antibody can bind the first one in multiple copies. So that now, for each antibody, black antibody, binding the antigen, you have 3, in this case, fluorochromos, for each reaction. So each antigen molecule is now marked by 3 fluorochromes instead of 1. 3 is just a plausible number, it's not really every time the number is different. So this is what you get. You see, the background is nearly identical in the two situations, but here the specific fluorescence is more defined, stronger. So the contrast between the specific and non-specific signal is better. The signal to noise ratio has been increased. So using indirect immunofluorescence compared with direct immunofluorescence increases the signal. The other possibility, which is usually employed in conjunction with this, with indirect immunofluorescence, is the opposite, is to lower the noise. How do we do that? Well, in part, the noise is due to the fact that the antibodies bind non-specifically to the bottom of the plate. So if we could prevent them from doing that, we will reduce the non-specific fluorescence, which constitutes the background of our experiment. When we do that, we have the same technique in indirect immunofluorescence, but this time we coat the plate where the cells are not with something, with a protein that basically blocks all possible sites to which the antibodies will bind non-specifically. The protein is not fluorescent, so it will not emit signal. And so, in this way, we prevent the antibody from binding non-specifically the plate. This also applies to the cells, but we don't want to make it complicated. There is also a non-specific binding of the antibody to the cells, so also that background is reduced. And the final outcome is this. Now the background is much lower, and the contrast between the cells and the background is pretty good. In this case, as I said, we had the background reduction, and we have combined the an increase in the signal with a decrease in background. Another possibility is this. Say our task is our task is to determine how many senescent cells we have in this culture. This is a culture of fibroblasts, and some of these cells are senescent. Now, senescent cells can be identified on morphological basis because they have characteristic shapes. But that's first imprecise. It's also subjective. Different observers will give you different results. So we need a better way to distinguish senescent from non-senescent cells in this culture. How do we do that? Well, we have to identify something that clearly distinguishes senescent cells from non-senescent cells. For many years we didn't have such a marker. Then it was found, one was found, and then a few others followed. One marker very commonly used is acidic beta-galactosidase activity. Beta-galactosidase, you probably know it as an enzyme expressed by E. coli, by Escherichia coli, which is the prototype of gene regulation discovered by Jacob de Monod. A similar activity is background. Let's see if we can do it. Antonio. I'll try to speak louder. So, acidic beta-galactosidase is a property of senescent cells. In fact, all cells have acidic beta-galactosidase activity, but senescent cells have it in higher amounts, so that if you stain the cells in a proper way, only the senescent cells will stay in blue. You can easily count them and detect them and determine the degree of senescence of the culture, for example, if you're interested in this. So, once again, you have found a way to distinguish the events of your interest, senescent cells, from the background of non-senescent cells. Once again, you've exploited the characteristic of the object you were looking for in order to detect it more easily. Yet another example is this. Suppose you need to count viruses. Now, viruses are very, very small. How do you count viruses? You take one, you put it here, you mark one. You take another one, you put it here, you mark another one, probably won't work. So how do you count them? Well, there are many ways to count viruses, including microscopic techniques, electron microscopic techniques, indirect techniques, really a wide variety. But there is an even more difficult task, that is, distinguish real viruses, real virions, from pseudo-virions. In Italian, pseudo-virion. What are pseudo-virions? You know that most viruses are assembled spontaneously. There is no model, there is no enzyme that assembles viruses. Simply you have a lot of viral proteins in the same place, in the same cell, and they spontaneously aggregate in the proper way and form a virus. Now, this mechanism, based on spontaneous diffusion of molecules, is prone to error, and in fact, typically a cell produces many-fold more pseudoviruses, pseudovirions, than really infectious virions. For example, I don't know which viruses are these. I can tell you that in the case of adenoviruses, for example, for each infectious virus produced by the cell, an infected cell typically produces 1,000 infectious viruses. The cell produces from 100 to 1,000 pseudovirions. So it's a very inefficient process. The inefficiency is overcome simply by the huge amount of viral proteins that eventually assemble a sufficient number of viruses for the propagation of the virus itself. Now, the difficult task I was mentioning is this. We don't only want to count viruses. We only want to count infectious viruses, excluding the pseudo-virions, which are sometimes obvious morphologically, so these are clearly defective viruses. But these others might also be defective. Perhaps the core of the virus might not be assembled properly. Perhaps they might not contain their nucleic acid. It's not obvious morphologically. So, how do we distinguish infectious? Listen to what I'm saying. This is the question for you. How do we distinguish infectious viruses from pseudovirions?
We infect something and see if it dies.
Okay,
like
yeah.
the plaque
Exactly. Good.
on a plate.
Right. Do you agree? Or a better idea? Or you want to improve on this idea? Everything is possible.
I don't know if it's possible to analyze the transcriptome of the virus to see if it's producing everything that it needs?
The virus only transcribes something once it infects the cell.
Mm.
So
Yes,
it is right
okay.
that an infectious virus eventually will transcribe something, but you need to infect the cell.
Okay.
You know the truth.
Mm.
Anything to add?
No.
So the idea is that. So of course the question was already pointing at the answer, but we in fact found a property that differentiates true viruses, infectious viruses, from non-infectious pseudo-variants. And so we exploited this property to count these infectious viruses. How do we do that? We make a plaque experiment. That is, in this case, they are probably phages. We have a lone plate uniformly plated with bacteria, and then we drop the viruses onto the plate. Each virus will kill the bacteria in the restricted area by counting the areas, the plaques. We can determine how many infectious viruses were in the preparation we are analyzing. So, in all circumstances, the trick is to identify or create, as in the case of Yamanaka, create something that we can either select for or easily distinguish on the background. So it's a matter of thinking sometimes deeply about the properties, the characteristics of the object we are interested in order to identify that specific feature that is useful for our purpose. Right, so that's your job when imagining a new experiment. How can I exploit the features of my object in order to select or identify? Good. Now, another possibility, again, we are looking for rare events. Say we are looking for a mutation. Say we are looking for a mutation that causes a disease, a monogenic disease. So a single mutation will cause that disease. How do we do that? It's in the next slide, but I would like you to. So many monogenic diseases now are well understood, and we know the gene that causes them. But sometimes we don't. Sometimes we haven't even described the disease, and so we don't know what causes it. So let's say somebody will takes to you a young child with a strange disease and says, it doesn't have a diagnosis, possibly because the disease is not described. Can you find the gene that causes the disease? Because you know from the family that this is probably a single gene disease. What would you do?
We need to sequenciare
Sequence.
the genome.
The genome of the child, yes?
Yes, of the patient, and make comparison analysis with a lot of human genomes to identify differences and then analyze them by one until, because of course there are some differences that are just variability,
No variations, yes.
and until we reach that one mutation, which is could be linked to the phenotype of the illness.
All right. In principle, that might work. That's basically the approach. The details need to be refined. In principle, that might work. And nowadays, now we have literally hundreds of thousands of human genomes completely or nearly completely sequenced. So you could compare your patient's genome with many, many other genomes. A better way is to compare the genome with the genome of the patient with that of his relatives. Because in these ways you would identify what mutations constantly segregate with the disease and what other variations are instead just casual. That's very efficient, and in fact nowadays it's very easy, comparatively speaking, to identify a single gene disease. So yes, that's a brute force approach. What does brute force means in this context? It means we are not trying to use a smart approach. We are not trying to think deeply as to what could tell us something about the identity of the gene we are looking for. Thing that geneticists were doing just 20, 15 years ago. There was no next generation sequencing back then. It was really hard work to identify a gene responsible for a disease. Now we simply take a brute force approach. We don't care. We sequence everything. And then we do a relatively simple comparison the way I described. So the brute force approach relies on having brute force. And in the case of next generation sequencing and of other current modern techniques, we do have force enough to really approach the problem we want to tackle. In the case of next generation sequencing, there are two technological revolutions that allow this brute force to be exerted. First is, of course, the ability to sequence millions, billions of nucleotides quite easily and quite fast and quite cheaply, which is also cool. And the other one is what? What else do we need? Pretty obvious. Once you have billions of what?
Database.
We need computers.
Okay.
There is no way to do sequences by hand, of course. So the fact that we do have available both incredible sequencing capabilities and unbelievable computing power allows us to use brute force in these types of approaches. And this is getting applied to ever more different research projects, approaches, techniques, etc. Really, this is become a workhorse for the scientists. Many problems are dramatically simplified by the availability of next generation sequencing. Because again, you don't have to smart out your object of interest. You just fish everything out, and in your net, you'll find the fish you're looking for. Another brute force approach is this. This approach has been applied to, in a specific case in the literature. This is just one of the thousands of examples I could have chosen. In a situation in which people were looking for a possible viral cause of a rare tumor called Merkel cell carcinoma. Merkel cell carcinoma is a skin carcinoma that the authors knew, of course, nearly always in immunodeficient patients. Patients that are immunodeficient for a variety of reasons, because of diseases, because of medical treatments, because of say organ transplantation and consequent immunosuppression, iatrogenic, caused by physicians, iatrogenic immunodeficiency, etc. Well, these people, these immunodeficient people, often develop this otherwise extremely rare tumor. So, the authors of this paper suspected that perhaps the tumor was caused by an infectious agent, which was able to infect only immunodeficient people, not immunocompromised. So they were now looking for something they didn't know for sure was there. They don't know virus. They didn't know any properties of this virus. They could guess. And they didn't even know whether the virus as such was in the tumor. Because in many instances, in many known instances of viral caused tumors, the virus can be no longer found in the tumor. There are only some remnants of the virus in the tumor. So how do you look for a virus of which you know nothing, including its own existence? How do you have a chance to find it? Well, this was really in the early days of next generation sequencing applications, these kinds of problems. You can see it from the number of reads, small sequences, say 50 nucleotide sequences that the sequencer were producing in the hundred thousands. Now these numbers will be billions. But still, this relatively weak approach was sufficient to do this. They sequenced the transcriptome of 4 cases of Merkel cell carcinoma. In one case, they sequenced a single carcinoma. In another case, they pooled together 3 tumors and sequenced them all together. Now, you might think this is strange. In science, they usually tell you that you should keep things separated, know exactly what you're doing, not mix everything together. But in fact, when you're looking for something you don't know, you try to balance your chances. You have a relatively, still at that time, not so powerful technique. And you didn't know in advance whether it was more, you have more chances of finding a potential virus by sequencing more deeply a single tumor, or sequencing 3 of them, hoping that at least one of them had the virus in it. So they did both. They then again combined the reads all together. They were capable of going back, of course. They had these 3 samples separated, stored, frozen, in case they needed to go back, so no worries. But they decided to work on them all together. They put the reads, essentially eliminated most of the reads, most of the sequences, because they were human. They were looking for something that was not human. So they simply aligned the sequences onto databases, onto human databases. Whatever did not align was a potential virus. One of these reads, only one, was indeed a viral sequence which the authors recognized was similar to a simian polyomavirus, which in non-human animals is a known tumor virus. At that time, no human polyomavirus was known to cause tumors in humans. Humans are typically more resistant to tumors than most humans, because we live so long. That's because we need to be resistant, otherwise we wouldn't be able to live so long. And so they found one of these sequences, from which then they reconstructed a long sequence by classical techniques, and reconstructed the whole genome of the virus. So once again, this was a brute force approach. They didn't try to divine, to guess, to imagine what kind of properties the virus might have. It was probably impossible or nearly impossible. They just adopted a brute force approach that allowed them eventually to find the virus. One final example. Finding a biomarker. What is a biomarker? The biomarker is an objectively detectable or measurable property which can be used as an indicator of normal or pathological states or response to therapeutic interventions. This is a complex definition that encompasses a lot of cases, but not all of them. It could be expanded even more. So basically it says a marker is whatever allows you to tell that this person is in a normal state, that this person has that disease, this person will respond or not to a given therapy. So you need to measure or to identify something in order to detect in what state the person is, or whether it will respond or not to a therapeutic approach. So do you know any biomarkers, according to the definition? Obviously.
Globuli bianchi,
Yes.
the number.
The common medical analysis we all do. Those are biomarkers. If you have high glucose in your blood, most likely you have diabetes. If you have low white cell counts, you probably have glucocytopenia of some kind, etc., etc., etc. So, the problem at hand in this case was to identify biomarkers for a rare disease, Creutzfeldt-Jakob disease. That is a devastating dementia that sometimes has a genetic origin or sometimes is sporadic. Genetic Creutzfeldt-Jakob disease usually strikes families that are known to carry the disease, and so it's not difficult to identify because you know what you're looking for, you know what mutations determine the disease. On the other hand, sporadic disease can strike any of us, although it's very, very, very rare. Problem is that there is no diagnosing the disease in life. You only diagnose the disease for sure after death, which of course, unfortunately, very early. Usually, between the diagnosis and the death, there's only a 6-month period. So, then what is the problem? Because there is no cure. So where would you identify the disease? Where would you want to identify for sure the disease? Well, first of all, at least to say something to the patients and to his relatives. But the more important problem is that these people might harbor the disease and be blood donors. If they donate blood to other people, they might infect them. Because the disease is caused by a prion. So, the problem at hand here was to find a reliable biomarker for Creutzfeldt-Jakob disease before the symptoms were evident, which will, of course, prevent the patient from donating blood for transfusions. And so again, here a brute force approach was taken. The authors here measured by mass spectrometry all plasma proteins they could identify and measure and detect in sufficient amounts as to be able to measure them. And so they compared a group of 10 Creutzfeldt-Jakob disease patients, or at least presumed Creutzfeldt-Jakob disease patients, so they were alive, the diagnosis was not versus a single number of controls of patients, not of patients, of healthy people. They even compare Creutzfeldt-Jakob patients with other neurological patients, just to have a tighter control, something closer, in order to determine whether they could distinguish that particular neurological disease from others. And so, here, what is shown here on a log scale, you see, not this is a linear scale. The log scale is here. Okay, what they measured was the ratio of the amount of any protein they could identify and measure in Creutzfeldt-Jakob disease patient versus normal individuals. So, if the patients had the same amount of that protein as the normal individuals, normal controls, the ratio was 1, right? Equal amounts. Patient over control. On the other hand, if the patients had increases or decreases in the amounts and concentration of some proteins, the ratio was significantly different from that. But indeed, you can see from this graph that 6 should be 7. This should probably be right. Yes, it should be. 7 proteins were identified as increased on average tenfold in Creutzfeldt-Jakob patients compared to controls in a neurological interest. So again, the approach was blind, that is, the authors didn't know what they were looking for, and was a brute force approach. They took a powerful technique, mass spectrometry, applied to plasma proteins, and looked and measured everything they could until they found something that was different in the two groups. So, as I said, this is the one last example in today's lesson, and it restates once again that in order to detect rare events or elusive events that are difficult to detect, we need to find a strategy. A strategy that either selects for those events, a strategy that exploits specific characteristics of those events, a strategy that sometimes applies brute force to the whole field until we find what we're looking for. Many things, you can use any strategy that works. In science, valid science is what yields results. Even if the strategy is not very smart, even if the strategy was wrong to start with, if you start with the wrong strategy, if you start from wrong assumptions, but find something true, that's science as well. So, highlights from this lesson. You'll find these very same highlights at the end of each chapter in the book. Rare or elusive events can be detected by increasing the signal-to-noise ratio. Selection, wherever applicable, is a very powerful approach to detect rare elements in an experimental system. Selection. Next generation sequencing, as well as other techniques such as mass spectrometry, allows us to reveal genetic elements in otherwise unmanageable contexts. Now we're finished with this lesson. Before we go on to the next one, are there any questions or comments? Not necessarily. If you're all right, we can go on.
Maybe one. Before brute forcing things, there was like a protocol where everything was unique, and every time you have to use your intellect just to find the best way possible, and maybe the only way possible without brute forcing.
Well, most brute force techniques are relatively young.
Yeah.
Like 20 years old. Before then, there was no brute force, no applicable brute force. In most cases, you just had to be smart. You had to think deeply about your problem and find a way to solve it. It's slow, difficult, and hard. Now things are still slow, difficult, and hard, but we do more difficult things, things that we wouldn't even approach before. Alright, so we'll start this lesson today and finish it the next time. And this lesson and the next one will deal with measurements and measures. Specifically measurements and measures in this lesson and measurement instruments in the next lesson. So what's the difference between measurements and measures? It's the same as in Italian. Misurazioni e misure. Measurements are the processes necessary to obtain measures. Measures are quantities that we associate by the process of measuring with the object we're studying. I'll repeat this in Italian because it's critical to distinguish. Misurazioni sono gli atti che utilizziamo per produrre misure. Le misure sono la quantità che associamo a una determinata proprietà. For example, of course, measurement and measures are widespread in science as well in everyday life. We commonly measure lengths, here for example the diameter, weight, times, etc., etc., etc. Measure many things. But knowing what we do when we measure things is extremely important in science. If you don't know what you're doing by measuring, you probably will incur in awful errors that will really drive you off with. Because measuring in the proper way is tricky. You have to know what you're doing, you have to know, this will be repeated many, many times, you have to know the power of your technique, and you have to know the limitations of your technique, of the kind of measurement you are using. Otherwise, you make mistakes. So, we can distinguish measurements into three classes. The first one is semi-quantitative evaluation. It's not really properly a measurement, it's rather an evaluation. It's still useful. How do we define it? We say that a semi-quantitative evaluation is a non-numerical evaluation of the variable of interest, which can be placed on a scale. Now, those are obscure. I'll make an example which I think will make you understand. This guy here is estimating, is evaluating the distances of 2 points from himself. He clearly sees that this point is closer to him than this one. He can't say what exactly is the distance between him and either of the 2 points. But he can say that this is closer than this. Now this is already important because based on this evaluation, he can say that if he has to pick up something that it is on this point as well as on this other point, he'd better pick it up here because it's closer, it takes a smaller effort. So it really has practical consequences. So if you like better ways you can resort to semi-quantitative evaluation, which will still give you some information that can be useful in your scientific endeavors. For example, to make a biological or medical example, oftentimes pathologists looking at slides or at sections of tissues make a semi-quantitative judgment. They say, for example, well, the tumor, in this section, the tumor covers about 60% of the area. This can be done with instruments, but sometimes the pathologists just do it by eye. You can say, for example, otherwise, that say if you're looking at an immunofluorescence or immunohistochemistry, that the fluorescence or histochemistry is either weak, stronger, strong, or very strong. So again, you can put it on a scale. Weak is clearly less than stronger. Stronger is clearly much less than very strong. So you are not implying that the distance between these various classes is the same. You just say that this comes first and this comes second. If you say, for example, to distinguish tall from average and small people, we are not saying that the difference between the average and the small is the same as the difference between the average and the tall people. We're just saying that these are smaller, these are taller yet. Okay? And again, this is useful. If you're looking for basket players, you select your population for the taller people. If you're looking for submarine mariners, you better pick the tall ones. Okay, so semi-quantitative approach. Next, we have relative quantitative measurement. This is a real measurement, often used in practice. Relative quantitative measurement, set down here, provides a numerical ratio. Here we have numbers, and the numbers are a ratio of the variable of interest in two or more objects, although it does not generate absolute measures. So what does it mean? This is an example. We can tell that this larger ball weighs exactly twice as each small ball. Okay. We don't know the weight of any of the balls here, but we can compare them. We can say that this one weighs twice as much as this. And this is very useful because, particularly in biology, most often, we are not interested in absolute measurements, but rather in variations. Oftentimes we say, this gene is expressed tenfold more in this situation than in this other. Or we say, the population of fish has decreased by 50%. How many were the fish before or after? We don't know exactly. But we know that if we take a sample, we now find 50% fewer fish than before, etc., etc., etc. Counting, by the way, is not exactly like measuring, but that's the detail we're not going. Finally, we have absolute quantitative measurement. In absolute quantitative measurement, we attribute to the variable of interest a numerical value associated to a unit of measurement. So, if we use a cylinder to measure the volume of some fluid, we get a number, say 100, associated to a unit of measurement, milliliters. All right, so is that clear enough? So in practice, we use, in science as well as in everyday life, all three kinds of measurements, depending on the circumstances, depending on what we are interested in, depending on the instruments, the methods, the capabilities we have at our disposal. In principle, you might be able to measure, I don't know, environmental radioactivity, but you don't have the instrument. So perhaps you could expose a film and determine how long it takes for the film to become black or gray. So that's a semi-quantitative evaluation, in which you say that in this environment it takes, say, 10 days, and in that other environment it takes 50 days. So there is more radioactivity here than there. You don't know how much, but still it is something. It tells you that that environment is a safer place than this one. If you use relative quantitation, again, you are basically looking for increases or decreases in a variable. And you have a number. You know how many fold that variable has increased or decreased between two experimental points or two time points or whatever. With absolute quantitative measurements, instead, you have what the name says, absolute measurements, which is a number of units of measurement. All right. Now, this is something that confuses a bit, but I want to say it anyway, because it is conceptually, at least, relevant. Even absolute quantitative measurements, in reality, are ratios, like relative measurements. The denominator is implied. It's not stated, but it's there. When we say that this sack here weighs 5 kilograms, it means 5-fold the kilogram of reference. So we never mentioned this, but it's there. And where is it? It's in the unit of measurements. When we say kilograms, we need to know what a kilogram is. The kilogram is the denominator of this absolute measurement. By the way, this was, since 2019, this is no longer the kilogram. This was the last human artifact used for as a unit of measurement. Since 2019, the unit of measurement is defined in a very complex way based on the Planck constant. And that's because we now define all standard units of measurement according to fundamental nature variables, fundamental nature quantities. But anyway, until 2019, that is just a few years ago, the denominator was this. Now the denominator is different, but it's still there. When we use a unit of measurement, we imply that we take a single unit of measure as the denominator or whatever we are measuring. Assuming that this is clear enough, and if it is not, please say, let's look at an application of these concepts to a practical, frequent actually, occurrence in the laboratory. Let's say we have a series of Western blots like these. So we are staining the gels for two proteins, HSP72, HSC70, and this is a technical further detection data acting which serves to normalize the bands, the lanes. We won't go into this. Let's just compare these bands. Okay, first of all, we could do a semi-quantitative evaluation. So, if we were to do a semi-quantitative evaluation of this Western, What would you say? Describe.
That we have the the biggest how do you say, band at 24 hours, and the smallest at 48, and I would say at 72 is the second smaller, and 96 the second bigger. I can put it on a scale. It's like the smallest is 48, 72 contrast,
Yeah.
and then 96 and 24 they're kind of similar.
Perfect. By the way, you have a very nice American pronunciation. Okay.
I watch a lot of TV series in English.
Okay, so and we will describe this. Supposing this is the same culture that we follow all the time, so the culture of cells. You can say that 24 hours we have an increase of expression in the expression of each sp2, which then falls down at 42 hours and then increases again up until at least 96 hours after the treatment or whatever happens to these cells. So this is a semi-quantitative evaluation, but it's still done in the laboratory. Now it's less common, but not really a few years ago. Most of the times people were evaluating their Western blots by holding the film against the window and saying, this is approximately threefold as large as this. And that was it, as far as quantitation was concerned. So, we can do something better. How would you convert somewhere, right?
Yes.
Somewhere, how would you convert this into a relative quantitation? Now you have to have numbers. Any ideas?
Areas, maybe.
Areas is not a bad idea.
Or density,
Density, yes.
which is similar to area because it's volume, mass, so something.
Right.
Uh,
Have you thought of this right now, or you had previous experience with Westerns?
no, I'm thinking about what property can I measure about that.
Right.
So-
Very good.
I had no idea.
And I don't know.
If I had asked you, I think you have come up with something at least close to this. Because once you're stimulated, you try to think. If you're just listening to him, you say, well. Alright, so yes, indeed, people are talking really about volume of the bands, which includes both the two obvious dimensions, length and width, and the third dimension, perpendicular to the slide here, which is density. So if you combine area with density, the third dimension, you get the volume of the band. And of course, both the area and the density are important in order to estimate the amount of protein, because this band here has an area fairly comparable with this, but it's much, much lighter. So you have to consider both the area and the density. And how do we do that in practice? Do you know by any chance?
Optical density and how much light it stops.
Yes. By what instrument?
Spectrophotometer.
Yeah, you could use a spectrophotometer, it would be a bit hard.
Okay.
Perhaps you know it, perhaps you don't.
No, spy spectra.
If you don't know, there is no guessing. It's called the densitometer.
Okay, we didn't know.
It's basically a scanner.
Okay.
There are now more modern ways to do this, but I think this example really tells you how things work. So the scanner measures the light that passes through a band. It's easy to measure the area of the band. The density is given by the amount of light that is capable of going through the band. So the light comes from the bottom part of the instrument, it is detected in the upper part. And so you have a number associated with each band, which is its volume, given by the multiplication of the area by the density. And so you get something like this. The volume of this band is on a relative scale. We are talking about relative quantification here. So we don't really know what a certain density of volume corresponds to how many nanograms, picograms of a protein. We don't know. But we do know that these two bands have the same volume. Now, you would say that they don't really have the same volume. This has a smaller area and possibly a smaller density, right? But this is where this lane comes into play. This is a control band
For the instrument.
for loading the gel lanes in the Western. So because beta-actin is supposed not to ever vary in any condition, which is not true, but it's an approximation, if you see a smaller band of beta-actin here, you have to proportionally increase this area here, this volume here, because you know that you have underloaded this line. The way it is done in practice is to divide this number here, this volume here, by this one. So never mind these calculations. Basically, these are the results you get. In the first lane, the gray columns show that the first two bands have a similar volume, that the third one has probably 1/10 or less of the previous two, the second 2 or 3 times the third, and the final one is pretty close to the first two ones. And these are all ratios. We give a number, we give a value to any one of these columns and compare the others to them. For example, for simplicity, often we label as one of the columns, say the control one. At the starting point, the value is 1, and the others are tenfold, 1/4, 1/10, etc. Same for the second one. Alright, so we have converted now this situation here, a semi-quantitative evaluation, into a relative quantitation here, in which we have a ratio. This over this is about 10. This over this is about 3. Of course, you know exactly what the ratio is. It's not shown. Alright, now the most difficult part, to convert a semi-quantitative evaluation into an absolute. That is, I want to know how many nanograms or picograms of protein are present in these bands. How would you do that? Women first? Problem is, you have an unknown amount of protein. You have to find a way to have a known denominator, a measurement unit, something known, in order to quantitate this.
We will need to know one of the bands for sure. Like if we put in the southern block
Western blot.
in the western block a protein with a known weight in nanograms, we then the relative density will show us how much the
An
other
absolute
bands.
measurement.
Yes.
Yes. Do you agree, Samuel?
Yeah.
Okay. Now, if I'd asked someone the first, you would have said I would have never guessed this. So try to be, more. Oops. Oh, yes. I don't know why. All right, so here it is. The way you do it is usually here is a different Western blot, which we are quantitating. This is an actual experiment from my laboratory. We are quantitating a protein called P21 in two conditions: proliferative versus quiescent cells. Never mind what they are. But we want to know not only that this is clearly smaller than this one, we don't only want to know it in a semi-quantitative way. Say this is perhaps 5 times less than the control one, than the proliferating band. We want to know how much P21 protein is in that band, which we can then use, if necessary, to calculate, say, how much protein is in the whole culture on an individual cell, if necessary. So, according to what Valeria said, we can have a reference, something in which we know the amount of protein that corresponds to the band we observe. And in fact, since we want to be very accurate in our determination, we have a single band. We have 7 containing different amounts in nanograms, yes, of the P21 protein. Now, these migrate more slowly than this one in the gel because they are recombinant proteins. So they have a tag that allows to purify them. But that's a technical thing that doesn't merit discussion here. What we have here, though, is a reference scale with absolute known weight associated to different volumes of the band. Now the task is very easy because all you have to do is to measure the volume, area times density of these known bands by densitometry and put them on a graph. Now the known bands are the white dots. You can see that the white dots align nicely, though a bit approximately, on the line here. Which means that the density scales linearly with volume, if you wish, of the band, scales linearly with the amount of protein, at least within a certain range. This is not infinite. Down here, we're already reaching the limit. The two dots are close to one another, to each other, because they are close to 0. These are close to the sensitivity of the assay. We barely see this band, so it's not surprising that 2 dots are close. But you see that they align pretty well on a single line. And we'll see when we talk about statistics what this line is. It's a linear regression, just to anticipate. But then the interesting part comes. You take the measured volume of this band and you put it on the graph. The volume is here. Here is the dot. The dot corresponds to 210, say, arbitrary units, or volume, whatever they are. But you can make it correspond to a specific amount of the protein, something like 1.8 nanograms. P21 here, something like 0.5 nanograms of P21 here. So this is pretty satisfactory in that we have now transformed all the way a semi-quantitative evaluation into an absolutely quantitative, And we can do it over and over again. For example, let's say I want to know the width of this table and this chair in a semi-quantitative fashion. What would you say?
You want to know the weight?
No, the width. Width. Like it.
Ah, okay. In a semi-quantitative, if you have more than one chair, like put how many?
That would be already a relative quantitation. I'm talking about semi-quantitative, the crudest.
Okay, just which is larger.
And smaller. Yes, wider.
Just look and
Look and say that the table is wider than
the chair. Yeah.
Right. Then comes Valeria and says, what does Valeria say?
I'm gonna put the chair like to one edge of the table and then move it to see if it's like twice.
How many times
yes.
it fits the table.
Yes.
Right. And it is roughly what? 2.5. So the chair now in a relative quantitative fashion, let's say it's easier. The table is 2.5 fold wider than the chair. Pretty good, because it says you can accommodate 2 chairs and not 3 under this table. It's a use of information. Now we want to know exactly how wide both the table and the chair are.
We measure.
We just measure them. In order to have an absolute quantitation, we need to have a measurement instrument suitable for the quantitation that we want to perform. Yes, so we need a meter in order to measure them. All right. So, again, another exercise of absolute quantitation, again coming from my lab. These are simple examples intentionally. I will give you very elaborate things to work on, but the principles are always the same. So it is convenient to think of simple examples and then simply extrapolate, simply generalize your understanding to more difficult situations. So here is another situation. Specifically, what were we doing here? We were using an ancient technique, because we were forced. More modern technique wouldn't work. So we resorted to an an old technique, no longer in use, basically, in the laboratory, which is cesium chloride separation. Cesium chloride separation is an ultracentrifugation technique that allows to separate DNA molecules, DNA filaments.
Linear from circular. No.
Yeah, it can
like
be used for
plasmid.
Yes, for plasmid preparation, correct. It was used to do, now no longer people do that thing. We have simple columns that we pass the plasmid through and you purify. But that's in fact was used for purifying plasmids. It's also capable of purifying molecules of DNA with different density. So, if you have, for example, DNA, regular DNA, it has a certain density. If you have DNA in which one helix, not the other, has been substituted with bromine deoxyuridine, which is heavier than regular vanillin, the density of the DNA will be higher and will, when ultracentrifuge on cesium chloride, in a cesium chloride solution, will stratify here. And if both helices are you do a substitute, you make it even heavier, and you stratify at the bottom of the tube. Again, never mind the technical details. What matters is that we needed to quantitate, absolutely quantitate the amount of DNA that we were able to recover from this preparation. How do we did that? How did we recover DNA from this? We basically punched a hole at the bottom of the tube and then collected drops of solution coming down one by one. So first this DNA came out, then this, right? So we have a number of tubes, each one with one or a few drops in them. But then we wanted to know how much DNA was in every one of those drops. So again, we used the trick suggested by Valeria. We had a reference here with known amounts of DNA. Oh, what is this? This is a petri dish with a gel put at the bottom of it. The same kind of gel that you use to run a DNA gel, to run DNA, to separate DNA. But in this case, it's just sitting at the bottom of the plate. And then you add a drop, a 1 microliter drop of a solution with a known concentration of DNA on top of the gel. The gel contains a DNA stain that produces fluorescence. So, increasing amounts, I don't know if you can read the numbers, I can't.
No, it's difficult to read the
Right,
number.
so not a good projection. Usually
10, 25, 50, 100,
right.
150.
Okay, so this is increasing amounts of DNA. Nanograms per microliter of solution. Then we simply take 1 microliter out of each of our little tubes, the fractions, so-called fractions from this tube, and quantitate them one by one. Basically, we compare by eye the intensity of this with these. It can be done in a more objective way, but this is good enough for our purposes. In fact, it is not that difficult to determine that these four drops here are in between these two, right? There is basically nothing in most drops. Here there is only a faint amount of DNA, and there is something else here which is not projected well. But anyway, you get the point. You have unknown amounts of DNA, but if you have a reference scale, you can fairly easily determine the exact amount. And so these drops are put on the plate in the same order in which they come out of the tube. So that is first drop, second drop, third drop, and so on. And so you can reconstruct the density of the whole tube by doing this. First drop, second drop, third drop, fourth drop, etc. And you have a nice graph showing that indeed you have reconstructed the three bands in the tube and the relative density and quantity of DNA contained in them. The quantity, in fact, is the area underneath each one of these curves. The integral of the curve to each. Good. So, this is what I wanted to say about the three classes of measurement. There are other ways to classify measurements, but I think this is one of the most useful in the laboratory. Now, after talking about measurements, we begin to discuss measures. Measures have a number of properties, some of which we'll discuss, many others we won't. We'll discuss the most important ones, the ones that are most useful in the laboratory, most important to know in the laboratory. Because, again, if you don't know what you're doing, you make horrible mistakes. I'll show you some examples in the next lesson. Okay, here is a table that I took from Nature Methods, a methodological journal. which deals with a number of properties, of characteristics of measures or measurements. Okay, here is a description of the property, and this is what you can do in order to assess that property or ameliorate your technique in order to better that property. All right, so let's start from two of these properties, which sound fairly similar: accuracy and precision. In Italian, accuratezza e precisione. Now, if you think of the words in Italian, they sound more or less the same. But in fact, in technical terms, they describe entirely different, separate, independent characteristics of measures. Okay, let's see the description. Accuracy. If a measurement is accurate, the measurement delivers the true value of the intended analyte. That is, we are measuring something of which we do know already the measure. A known standard. We have a known standard of which we know the measure. So we now use our instrument to determine that measure, and we determine how close our measures come to the actual measure. All right. So here we have a first problem that I'll let you think about. How do we know the real measure of the standard? That is, thinking of the last example I've shown you a couple of slides ago. How did I know how much DNA was present in each of the droplets that made up the reference scale on the paper?
I weighed it.
I had weighed it. Okay. How did I know that my scale, la mia bilancia, was accurate, was giving me the right weight? Somebody must have used another standard to make sure that the scale was accurate. How did he know that the standard he was using was accurate, was really weighed that much, say 1 gram or 1 milligram, or whatever?
The important is that are all the same, not.
Yeah, there is no way.
Yeah.
If we go backward like this, there's no going back to one original.
Exactly. So the bottom line is, you never know exactly what is the true measure. The best you can do is to get the best measure that you can. According to the best methods, the best instruments we have at our disposal, this is the measure of this object, of this property of this object. And that's it. There is no higher instance. There is no knowing better than that. Right, so this is important to know. A measure always contains an error. It's unsuppressible. Even if you had a perfect measure, you wouldn't know. Because if you repeated it, you wouldn't get the same numbers. Not exactly the same. So there is always a variation in a series of measurements of the same thing. So accuracy means keeping this variation as small as possible. The more this variation, the smaller this variation, the higher the accuracy of your measure. Okay, how do you get your accuracy as best as possible? Test your experimental observation using orthogonal analytical methods. We talked about orthogonal methods. If we use two methods to measure the same variable, they give you the same identical, very similar numbers, you are convinced that this is pretty close to the real measure, which you will never know. Exactly. And then, of course, use well-defined reference materials to check the instrument response and method validity. In fact, this is it's a very complicated thing to do. What is a well-referenced material? It's a material that you can trace back all the way to the definition of measurement unit. That is, in some laboratory, in some highly specialized laboratories in the world, they have measured that standard in the best possible way and determined how much it, say, weighs. Right? Then from that standard, they make copies, they distribute it to different countries. And then the measurement offices in those countries that make copies of them, making sure that the copies were as close as possible to their originals and so on. This way, you make sure that the world uses as close to the same measurement unit all the time. And this is very, very important because a measurement unit has a property that we haven't discussed when I first mentioned it. Just a second. Thank you very much. Se riesco a dire. Per favore, parlate piano. Grazie. Okay, so we have this chain that ensure we're using the same measurement unit all over the world. And in fact, as I was saying here, the measurement units usually have a property that they are accepted by a large number of people, by many countries. That's what makes them a real measurement unit. Even the length of my arm can be a measurement unit, but you wouldn't know what it is. Your mother wouldn't know what it is. Somebody in France wouldn't know what it is. So it is not usable. We have to have the same measurement units, if possible, for the whole world. There was a probe sent to Mars, I think it was like 12 years ago or so, in which, unknown to the people involved, part of the software calculated in meters and another part in yards. The probe crashed onto Mars, of course, because they weren't using the same measurement units. So that tells you the importance of sticking to measurement units. All right, so time's out. So we'll come back to these properties next time. And I'm sure we'll have fun, because here is a moment in which we can really reason about what the meaning of what we do is and how we can do it best.